Swayam Prabha. Digital India. Educated India.
Hello, my dear students. Welcome to Swayam Prabha, sponsored by MHRD, New Delhi. Course title Medicinal Chemistry. Lecture title Principle of Drug Design, Part 2. Faculty name Dhamak Vikrant Murlidhar. Module number 6, lecture number 10. So we'll see firstly the contents of today's lecture. In part 1, we have completed some concepts. Now in part 2, so the first concept we will see, that is analog design. After that, category of analog design. After that, bioisosters and its classification. After that, rational drug design. And after that, prodrugs and its uses. So these two classes, that is analog design and rational drug design. So these are two important classes of drug designs. Bioisosters are such compounds or such groups which shows similar characters of a drug. And the concept of prodrug. So we'll see in detail types of prodrugs and its application. We'll see the first concept, that is analog design. Analog design is usually defined as the modification of drug molecule or any bioactive compounds in order to prepare a new molecule showing chemical and biological similarities with original model compound. So meaning of this thing is that once you come across in medicinal chemistry, any drug classification, so you will find that there is number of compounds in one class. So they are little bit modified. So modified means, so there is one bioactive compound, or we can say a lead compound, and a lead compound is chemically little bit modified. But like example, introduction of an phenyl ring, or phenyl ring, it has been substituted with halides and with different halides. So such kind of modifications are done with bioactive compounds, and it shows little bit similar or little bit higher activity than a lead compound, or little bit lower activity than the lead compound. So that thing is said to be an analog design, means designing of an same molecule with little bit modification. And in the last lecture we have seen the concept of a lead, and so the lead, it has been modified, and the series of compounds, it has been prepared. So we'll see an example of analog design also. And design of analogs make uses of procedures such as synthesis of homologous, as we have discussed it, that a series of compound, it has been prepared. Vinylogous, isosters. So in this lecture we will come across the concept of what is an isosters. After that, positional isomers. So it is one of a class from an isomerism, that is positional isomer, only change in functional group of an position. So that thing is said to be a positional isomer. After that, optical isomers, like enantiomers, we can say that your two enantiomers, either they have same activity or different activity. So depending on how it fits to end receptor. After the transformation of ring system, so sometimes instead of pyridine ring or instead of benzene ring, so other ring systems are been also utilized. So that is heterocyclic system and synthesis of twin drugs. So these are approaches how, or so these are the procedure how analog are been designed. So we'll see an example of an analog design. So this is an example of an tetracycline. So in the last lecture, we have seen that some antibiotics, they are being prepared by random screening before 1970s, and tetracycline is such an example. So it has been obtained from an random screening. So this tetracycline, so it is an basic moiety having 4 rings and at some position hydroxyl group and some position carbonyl group. So little bit modification in this 4 rings. So we'll see one by one. So here you can see that this is a structure of 6-deoxytetracycline. So instead of this OH, it has been replaced by H. After that, doxycycline, this OH, it has been replaced by H, and it is having one more OH. And after that, minocycline. So here you can see that here it is removal of this OH and CS3, and instead of that, it has been substituted by N-dimethyl. So from this, you can understand with this example of an tetracycline that the tetracycline, it is a basic lead moiety, and little bit modification they have been done. Either the potency of drug it has been increased, or either some pharmacokinetic parameters like adsorption, distribution, metabolism, and elimination. So there is different pattern of an little bit in this structure compared to the tetracycline, and many time it has been happened that by use of some different groups. So in the last lecture also we have seen that by use of some different groups like introducing some hydrophobic groups, so it will increase in lipophilicity, and by introducing polar group, so it will increase hydrophilicity but it will decrease lipophilicity. So it does depend on what kind of group we are introducing. So from this example, now you might have understood that what is actually an analog design. So name itself indicates that finding an similar analog to the parent compound and making little bit modification, and this modification leads to some important characteristics that we will see what are the applications of analog design also. So now we'll come towards what is importance of analog design. In the pharmaceutical industry, when a company launches a pioneer drug resulting from an original invention and corresponding to a real therapeutic need, or considerably before then, the competitors initiate immediately search for similar acting drugs in order to keep their market position. The analogs can be simple copies or display some improvements over the original drug molecules. So whenever any company it comes with any therapeutic drug which has been invented, so competitors from other industry, so they prepare a similar analog. So similar analog, it means that they change little bit modifications, or either they make a use of an isosters that we will see in the lecture. So what is that isosters? So by this way they prepare an analogs, and these analogs may display similar activities or maybe similar copies within chemical structure, or there may be improvement of an activities, or there may be improvement of an pharmacokinetic parameters like absorption, distribution, metabolism, and elimination. So due to which also it also impacts on a cost of drug. As this company, first company which has invented a drug, so the cost of manufacturing or cost of research, it has been increased due to the invention what company has invested for inventing a drug. But when other companies come, they make similar kind of drugs with an use of an analog design concept, and due to which the cost of drug also reduces. As other companies bring a similar kind of a drug in the market with having similar type of an potency, efficiency, and due to which the earlier company has to reduce the cost of a drug, and due to which the cost of drug also reduces in the market. So this is an importance of an analog design. Now we'll come towards some categories of analog design. Analog presenting chemical and pharmacological similarity. After that, analog presenting only chemical similarity. And after that, analog displaying similar pharmacological properties but presenting totally different chemical structures. So first category, that is analog presenting chemical and pharmacological similarity. So the meaning of this thing is that chemical structure is also similar, as well as an pharmacological structure, pharmacological activity of drug is also similar. Second category, that is analog presenting only chemical similarity. So the difference between this two category, it seems to be that chemically structures or structures are similar but the pharmacological activity for second one is different one. After that, the third category, that is pharmacological properties but presenting totally different chemical structures. So from this it can be understood that pharmacological activities are similar but chemical structures are different. So now we'll see each class with an example. So the first class, that is analogs presenting chemical and pharmacological similarity. These analogs correspond to the class of drugs often referred as Me Too drugs. Usually they are improved versions of pioneer drug over which they present a pharmacological, pharmacodynamic or biopharmaceutical advantage. So analog design, so importance of analog design is also one thing, that over an lead molecule or over an pioneer drug, so it improves in pharmacological, pharmacodynamic and biopharmaceutical advantages. So it changes and some parameters like ADME, also it improves and some pharmacodynamic effects, that is mean potency and efficacy of a drug, and also interaction with an receptor of a drug molecule. So there are little bit modification in the drug molecule which improves and activity of a drug. So looking towards example of this class, that is AC inhibitor, angiotensin converting enzyme. So captopril, it is one of a lead molecule, and this lead molecule now it has been converted having similar basic structure. So this is structure of lisinopril. So chemically they are similar. So here you can see that a similar kind of a ring it is having, and after that there are little bit modification on a drug molecule. So these modifications are usually for increasing pharmacodynamic properties or some biopharmaceutical, taking some biopharmaceutical advantage, that is pharmacokinetic parameters. but the basic moieties are similar. And captopril, it is one of a lead molecule, and lisinopril, enalapril. So all prils, they are been analogs of an captopril. After that, second class, that is analog presenting only chemical similarities. The term structural analogs contains a compound originally prepared as close and patentable analogs of novel lead, but for which biological assay revealed totally unexpected pharmacological properties. So lead molecule or patentable drug, similar kind of structural analogs are being prepared by some isosteric replacement or by not disturbing electronic parameter as well as in steric parameters. So similar kind of drugs are being prepared, but with little modification, but the pharmacological activity, it is different. And due to the difference in pharmacological activity, the use of that drug becomes totally different. Now we'll see an example of this class, the discovery of antidepressant properties of imipramine, which was originally designed as an analog of potent neuroleptic drug, that is clomipramine. So it is having just only a little bit difference, that it is having an chlorine substituted instead of an hydrogen on this aromatic ring. So this is structure of an imipramine, and imipramine, it is having an antidepressant activities, while clomipramine, it is having an neuroleptic activity. Both are actually CNS depressant, but activities are a little bit different, and due to which their uses are different. After that, analogs displaying similar pharmacological properties but presenting totally different chemical structure. The term functional analogs for such compound. So such compounds are also called functional analogs, because the functions are similar, so it is said to be an functional analogs. Example, neuroleptic chlorpromazine and haloperidol. They show different affinities for dopamine and benzodiazepine receptors. So this is a structure of an chlorpromazine having an chlorine substitution, and second class, that is haloperidol. So this haloperidol, so here you can see that totally structures are different, but the activity, it is similar one. Like same in the example of an dopamine and benzodiazepine. So these are having a different structures, benzodiazepine and dopamine, it is one of a neurotransmitter, but the activities, they are similar, that is they are anti CNS, that is CNS depressant. So these are the three classes of an analog design. After that, we will come towards a concept of an bioisosters. Substituents or group with chemical or physical similarity that produce similar biological properties. So here the compounds, they are having similar type of substituents, and they produce similar biological properties also. Isosteric replacement requires substitution of one atom or a group of atoms in a parent compound by another with similar electronic and steric configuration. So it is important thing that these two parameters, that is electronic and steric configuration of a drug, it should not change. So they should remain similar, even though there is isosteric replacement, but these two parameters should not change. And change or replacement or substitution. So it is of just an atom or it is group of an atom in a parent compound. So parent compound here it means a lead compound. So lead compound it has been little bit modified, and after that it shows similar kind of an biological properties. So what is need of an bioisosteric replacement? So that is for improved selectivity or greater selectivity of a drug. When drug it interacts with an receptor, at that time with help of this replacement of a group or an atom, so there may be a chance of an improving and selectivity towards an receptor. Less side effect. So in earlier lecture also we have seen some drugs which were having similar chemical structures, but they were having little bit modification, and the side effects of drug molecule it was been changed. After that, decrease toxicity. So toxicity of the side effects that relates with also an toxicity. That toxicity is also been reduced by changing a little bit modification in the drug molecule. After that, improved pharmacokinetics. So it is one of an important thing that by improving an pharmacokinetic of a drug, activity of a drug, it increases. So one of an example also in earlier lecture we have discussed that ciprofloxacin, or we can say fluoroquinolone. Ciprofloxacin actually it is a drug from a fluoroquinolone class. So when fluorine it was been introduced at 6th position instead of an hydrogen atom, the lipophilicity of a molecule was been increased of quinolones, and it turned to an fluoroquinolone. And after that it shown that even though similar atoms like fluorine or similar halogens like chlorine, bromine, iodine, they didn't show similar type of an activity or pharmacokinetic properties within fluorine. So the class it was been developed that was fluoroquinolone. So instead of hydrogen it was been replaced by an fluorine group. After that, increased stability. So when a drug it has been susceptible for simple reactions such as an oxidation, so groups they are been converted into, or isosters they are been replaced, or isosteric groups they are been replaced in such a way that they are not prone to an oxidation. After that, there are some more advantages of an biosteric replacement. Size, shape, electronic effect, lipid solubility, water solubility, pK, chemical reactivity, hydrogen bonding are the parameters that influences potency, selectivity, and duration of an action. So in the earlier lectures also we have studied this. What are the things that is lipid solubility, water solubility. When some lipid or hydrophobic bioesters are been replaced, so it improves an lipid solubility. After that, water solubility is also been improved by using some ionic bioesters. Or after that, pK, so it all depends on an ionization of a drug. After that, chemical reactivity. So chemical reactivity in sense it is for an chemical interaction with receptor of a drug moiety. After that, hydrogen bonding. So similar kind of thing, that chemical reactivity when it interacts with a receptor. So there is importance of an hydrogen bonding also, because we have earlier also seen such kind of an interactions they are present in drug receptor interactions. And all these parameters, also size and shape of a molecule and electronic effect, these also influences potency and selectivity and duration of an action. So simple example like drug when it is been esterified, so duration of an action it increases because drug has to convert into an carboxylic from an ester, and after that it will get ionized and it will show an uh, activity. So these are some advantages on bioesters. And also bioisosterism is effective because it affects above parameter to less or more extent. So either it will increase potency, selectivity and duration of an action, and all these things it depends on these parameters. So there now we'll come towards an point classification of bioisosterism. So there are two main classes of an bioisosterism. First class, that is classical bioisosters, and second class, that is non-classical bioisosters. So in case of an classical bioisosters, so electronic parameter and steric parameter it does not changes. So there are some classes of an classical bioisters, that is first class, that is univalent atoms or groups like carbon, nitrogen, oxygen, sulfur, chlorine, bromine. So univalent it means that there is replacement of just only one atom, and replacement of one atom after that bivalent atom or groups. So bivalent it means that the molecule it is or atom it is having and two valencies. So like ethers, sulfur and amide or amine groups. After that trivalent, so CH it is sometimes it has been replaced by this N. After that phosphorus, after that substituted amine. After that tetravalent atoms, that is having four bonds, the carbon it has been replaced, nitrogen, quaternary ammonium we can say it has been replaced, after that phosphorus. So these are some classes of an classical bioisosters. Now we'll see an examples of an classical univalent bioisosters. 5-fluorouracil, a thymidylate synthase inhibitor, interrupting actions of this enzyme blocks synthesis of an pyrimidine thymidine synthesis, which is a nucleoside requirement for an DNA replication. So in the earlier lectures we have seen the structures of DNA, that they contain purine and pyrimidine analogs, and this thymidine it is an pyrimidine analog, and this thymidylate synthase it is one of an enzyme which is required for synthesis of this pyrimidine analog, that is thymidine. And 5-fluorouracil it blocks an action of this enzyme which is required for DNA replication further. So this 5-fluorouracil, so it was been designed from an uracil. So just there is replacement of an hydrogen, and this replacement of an hydrogen it is done with the help of an fluorine, and fluorine it has been replaced with the help of an hydrogen, and it has improved the lipophilicity of a molecule because fluorine it improves in lipophilicity, and due to which potency or the action or biological effect of a molecule has been increased. And why it is said to be an univalent? Because only one atom having an one valency, like hydrogen is having one valency and fluorine is having one valency, or they are been connected with only one bond, so they are classified under univalent class. After that, classical bivalent bioisosters. Procanamide is an amide, has longer duration of action than procan. An ester, because of isosteric replacement of ester oxygen with a nitrogen atom. So these are an example of an procanamide and procan. So here you can see that NH, it is replaced by an oxygen. So divalent atom, it is replaced by an oxygen, that is divalent atom oxygen, and due to which there is improved in action, that is it is having a longer duration of an action, because for an hydrolysis it requires some time compared to an ester. After that we will come to an now second class of an bioisosterism. So first class we seen of an classical bioisosters. After that second class, that is non-classical bioisosters. So non-classical do not have similar number of atoms and do not fit steric and electronic rules of classical isosters, but they produce similar biological activity. So though they are having different electronic and steric parameters, still they produce similar biological activities. So example like estradiol. So here you can see the structure, it is not very similar, and there is removal of these two rings and it has been replaced by an open chain structure. And biological activity of this diethyl stilbestrol is improved than estradiol, and just a ring they have been opened and CH2 over here, it is now free, and still it is showing a similar biological activity. Also this penta ring, pentahydroxyl ring, it has been replaced by an phenol, and still the activity it has been improved. So from this you can understand that. Also you can see that steric and electronic parameters, they are different. So here it is an aromatic ring and here it is an cyclopentane ring, and it is having high degree of an unsaturation, and this ring completely it has been saturated one. Now we'll come towards the next concept, that is rational drug design. So there are two classes of actually in drug design. So one is analog drug design and second one is rational drug design. It is a process in which finding of new medication based on knowledge of biological target it is done. So like example of an nowadays computer aided drug design, it has been used tremendously and vastly. So in case of an computer aided drug design, already the biological target it has been studied or it has been known, and the protein structure of that biological target it is known, and this protein structure it is kept in protein data bank online, and from which by using some softwares like Schrodinger or some softwares related with an computer aided drug design, so this make use of an virtual designing of a drug, and this virtual designing of a drug it has been suited with a biological target, and after that measuring of a score function, and which shows a major score that drugs are been synthesized. So it is one of a way in the rational drug design. Basically in rational drug design, biological targets are identified, on based on that a new drug molecule it has been invented. It involves designing of small molecules that are complementary in shape and charge to biomolecular target. Like neurotransmitters are also important in such case of an rational drug design, that with the help of neurotransmitter one can make and drugs having similar size and shape. So we'll come across an example of an rational drug design. Rational drug design begins with the hypothesis that modulation of specific biological target may have therapeutic value, and it is based on hypothesis that targeting and that biological activity of an specific organ, and which induces an therapeutic value later on. So by targeting that biological organ or biological target, so one can stop the activity over there, or it can improve the activity over there, and due to which the therapeutic value of a drug it improves. So now we'll come towards steps in rational drug design. So what are the basic steps in rational drug design? So first step is that identify a receptor or enzyme that is relevant to a disease they are going to design a drug for. Firstly, identifying enzyme and receptor which is responsible for that disease. After that, elucidation of the structure and function of that receptor and enzyme. So it is done by some spectroscopic techniques such as NMR. The structure it has been identified, and once structure it has been identified, so one can understand the interaction of a drug at an active site. After that, third step, that is and last step, use the information from step 2 in order to design a drug molecule that interacts with the receptor or enzyme in a therapeutically beneficial way. So these are the 3 steps which are being utilized in the rational drug design. So already I have discussed that there are 2 ways for a drug design. So one is analog design and second one is a rational drug design. And in case of a rational drug design, so firstly a target it has been identified, and once target it has been identified, and after that only a drug molecule it has been invented for a therapeutic purpose. And in case of an analog design, so from a lead molecule, other molecules are been designed with an homologous series, having little bit modification with the help of an bioisosteres, or introducing some new groups or new chains or new aromatic rings or heterocyclic rings to improve and pharmacodynamic and kinetic parameter. Now we'll see an example of a rational drug design. So example, it is cimetidine. So in case of a rational drug design, firstly we need to identify the problem, and once we identify a problem, so the problem is that histamine triggers a release of stomach acid. Want a histamine antagonist to prevent stomach acid release by histamine. So in case of a rational drug design, firstly problem it has been identified, and what are the ways that create a problem, and these ways are being inhibited or they are being stopped, and this is how it works. So here the problem is that histamine, when it has been released, so it releases an acid in the stomach, or it causes an ulcer, and therefore we need to prevent this acid release. And this acid release, it is responsible, so the neurotransmitter that is histamine which is responsible, and once this histamine it has been blocked or it has been antagonized, so there will be no release of an acid in the stomach. So in case of a rational drug design, so drug design for that year, the help of an histamine it has been taken, and histamine analogs were synthesized with systemically varied structure, and out of that N-guanyl histamine showed some antagonistic properties. So now that became a lead molecule for this class. So it is now an antihistaminic drug. Histamines are agonist and antihistamines are antagonist. So little bit modification in the neurotransmitter, it creates an antagonist, like in case of an acetylcholine also. So acetylcholine, it is one of a neurotransmitter for an cholinergic system. By change or little bit modification of an structure, so it becomes an antagonist. So here we will see how it was been developed. So this is a structure of an histamine, and histamine it has been inserted by an guanyl group in histamine, and this guanyl showed some antagonistic properties, and due to which this antagonistic properties, so there were some modifications made on this, and it served as a lead molecule, and as it serves as a lead molecule for further antagonist, so here a potent metamide it was been synthesized. So instead of this nitrogen, it was been replaced by an sulfur. So it is divalent bioisoster, and it has been replaced with an sulfur having a substitution of an methyl group. But the problem with this drug, it was potent but toxic, and after that cimetidine it was been synthesized. So cimetidine sulfur it was been replaced with the help of an nitrogen and with cyano group attached to it, and as now it is forming having an antagonistic properties, and this ethyl chain it is also been replaced with an sulfur, or by introducing an sulfur and adding one more methyl in it, and it has formed an cimetidine. So this is how, from example, you can understand how rational drug designs are been done. So in case of an rational drug design, it is little bit different from an analog design. That in case of an rational drug design, firstly biological targets are been identified, and what are the functions of that biological target related with that disease. So that ways or pathways they are been blocked. So same thing has been happened over here, that this histamine was responsible for release of an acid into an stomach, and to prevent ulcer, so antagonist has to be synthesized, and so the antagonist it is been synthesized with the help of neurotransmitter itself, and this neurotransmitter that is histamine, and making little bit changes with an histamine. So introducing an guanidine in the histamine, it become a lead molecule, and from that lead molecule there were some modifications, there were some additions of some groups or some atoms, and which lead to an potent cimetidine molecule. Also metamide was potent, but it was toxic. Now we'll come towards the next concept, that is prodrug. Prodrug is a medication or a compound that after administration is metabolized and converted within the body, and now it becomes a pharmacological active. So there are four processes in case of an pharmacokinetic, that is absorption, distribution, metabolism, and elimination. So this prodrug concept, a drug after metabolism it becomes active. Actually at the time of an metabolism, so there is introductions of some ionic group with the help of some enzymes like cytochrome phosphates, and with the help of that enzyme it has been converted to an ionic. So after it becomes an ionic, it has been eliminated. But the concept of an prodrug, it is that once drug it becomes an metabolite, so it becomes more active or it becomes more interactive with a receptor. And once it becomes an active with a receptor, so there is an pharmacodynamic activity of the drug, and this is done because there are some limitation with directly introducing a drug, and to avoid this direct introduction of a drug, so this pro drug concept, it has been developed. So we'll see in an example how it works actually. So here you can see a drug molecule and which tries to cross an barrier. So one of an example I have taken over here, that the solubility barrier, it means that drug, it is more lipophilic and it is not able to go through some biological fluids in the body, and due to which it has been repelled. So now conjugation, it means that joining. Now drug, it has been joined with some molecule. Now it becomes an prodrug. So molecule is now ionic. Now we can say it is now an ionic in nature. So this part, it is an ionic, and this ionic nature of a drug molecule, it crosses an solubility parameter. As it crosses an solubility parameter, or as it crosses an membrane, or it goes through some biological fluids, so there is biotransformation. So biotransformation and metabolism, it is the same thing. That is nothing but alteration in the structure. So alteration in the structure means a drug molecule, it has been separated, and now this molecule which act as an pro for a drug, it has been now separated. So it acts just as a carrier for a drug to cross this membrane, and after metabolism, now drug is active one. So this drug, now it enters into a cell and it shows the activity towards a receptor. Now we'll come towards an application of an prodrug. Firstly, improvement of test. Some drugs, they are not very, they are so bitter, and in case of an pediatric, this thing happens that the pediatric patients, they are not able to take these bitter drugs, and for an improvement of an test, a prodrug concept, it is been utilized. Like example of an chloramphenicol. Chloramphenicol is an extremely bitter drug, has been derivatized to chloramphenicol palmitate. So palmitate, it is nothing but an ester derivative of an chloramphenicol, and this ester derivative of an chloramphenicol is now having a different taste. So it is less bitter compared to an chloramphenicol. After that, change in physical form of a drug. Sometimes it happens that drugs are not suitable, or they are prone to oxidize, or they are prone to some changes, and with a suitable form, either it may be solid, liquid, or gas, so drugs can be changed. Like trichloroethanol crystallize to a solid. So actually it is a liquid, so it can be converted to an crystalline form, and now it becomes unstable. In liquid form, it is not stable. Reduction in GI irritation. So example like aspirin. So it is one of a famous drug in market, that is aspirin, but it's an prodrug for an salicylic acid. So salicylic acid, name itself indicate that it is having an acid, and this acid causes an irritation in GI, and due to which it is converted to an aspirin. So it has been derivatized to an ester. So this acidic functional group converted to an ester, and which forms an prodrug, and once it enters into an gastric or in the stomach, so there is no irritation. And once it enter into an basic medium, that is once it enter into an colon, so there is our small intestine, so ester is converted to an acid, and as it is converted to an acid, so it becomes an active salicylic acid, and which is now an active for its pharmacodynamic effect. So it has been converted to from acid to ester because there is basic environment in colon, and this changes acid to ester. After that, reduction in pain on injection. So example like phenytoin, it is one of an antidepressant drug, and phosphenotoin, it is one of an prodrug for an phenytoin. So there will be reduction in the pain, and once it has been administered, so after that it convert into an phenytoin. So there are some more applications of an prodrug. After that, enhancement of drug solubility and dissolution rate. Estramoustin phosphate is a prodrug for estramoustin. So this phosphate, it is responsible for improving the solubility of a drug and also an dissolution rate. Alone estramoustin, it is not so soluble, and dissolution rates are also lower. After that, enhancement of chemical stability. After that, enhancement of bioavailability. Example, dopamine was made useful by making a precursor of L-dopa. See, this happened that dopamine, when it has been administered directly, so it does not causes an blood-brain barrier. As dopamine, it is required in the brain, so there is big hurdle of an barrier, that is blood-brain barrier, and due to which dopamine, it does not passes that blood-brain barrier. But as drug, it has been converted to L-dopa. So this L-dopa, when it has been administered, so it can pass an blood-brain barrier, and after that, enzymes over there, it converts L-dopa to dopamine, and now which becomes an active form and which can interact with a drug. Prevention of pre-systematic metabolism. Like heroin, it serves as a prodrug for an morphine. So heroin is nothing but a diacetyl derivative for an morphine, and this diacetyl derivative of an heroin does not allow and metabolism of a drug. So that is nothing but an like first pass effect. So it has been metabolized in the liver rather than going towards a receptor. Firstly, it has been metabolized. So it can be prevented. After that, prolongation duration of an action. Like example of an testosterone propionate and testosterone. Propionate, it is an example that it has been converted to an propane plus ester. It refers to an ester, and once this propionate, it has been introduced into an testosterone, so there will be prolongation duration of an action. Testosterone, it will not metabolize very easily, and the duration of an action, it will be for longer time. Now we'll come towards an types of prodrug. Carrier linked prodrug consists of an active drug covalently linked to an inert carrier or transport moiety, generally ester or amide. So we have seen an example of such type of an prodrugs also. So this is an active drug and inert carrier. So the carrier, it should not be active or it should not be toxic in nature. And after, so prodrug formation, so there will be linkage with an covalent bond, and this linkage, it can be cleaved after enzymatic process or chemical process in vivo or in the body. And now active form, it is free to act, and this inert carrier, so it has been eliminated. So here is an example: chloramphenicol succinate. Chloramphenicol succinate, it is one of an prodrug having an succinate over here, and it is converted to chloramphenicol with the help of an esterase enzyme. So it removes, it hydrolyzes and this carboxylic acid and ester over here, and now this OH, it is free, and due to which now active molecule, now it is free to act. After that, bioprecursor, so name itself indicate that, so these drugs are metabolized, and after that they serve as an active agent, and this metabolism is done with certain chemical reactions such as oxidation reduction in the body. Like example of an nabutane, it is used in non-steroidal anti-inflammatory, used in arthritis. After a series of oxidation and decarboxylation, so it forms an active form which inhibits biosynthesis of an cyclooxygenase and also prostaglandin. Now we'll see summary of this session. So in the first instance, we studied analog design, that is modification of drug molecule in order to prepare a new molecule showing chemical and biological similarities. So we have seen an example of an captopril. So it serves as an lead molecule. So lead molecule, it has been modified to prepare a series of an compound, and this series of compound shows biological similarities or biological activity, it is similar. After that category of analog design, same biological and chemical activity is first category. Structures are also same and activities are also same. After that, different chemical structure but activities are similar. And after that last category, it was that activity is same but structures are different. After that, we have seen a concept bioisoesters and its classification. So bioesters are substituents or group with chemical or physical similarities that produce similar biological effect. So we have discussed the example of an fluoroquinol. It has been replaced, fluorine was been replaced by an hydrogen, and due to which lipophilicity of a molecule was increased and activity of molecule, it was also increased. And its classification, there are two classes. One is classical and second one is non-classical. In case of an classical, electronic parameters remain similar and its configuration remains similar, but in case of an non-classical, electronic parameter does changes. And in case of an classical bioisoesters, we've seen 4 different classes. After that, a rational drug design, process in which finding of new medication based on knowledge of biological target is done. Also, we have discussed a thing that is computer aided drug design, in which biological targets are already known or they are been identified, and once biological target it is known, the pathways or the process for which disease or disorder causes, that it has been blocked with the use of drug. After that, we have seen the concept of prodrugs. After drug it has been metabolized, it converts to an active form. So we have seen certain examples and its application. Thank you. So in this session we have completed module number 6 and this was lecture number 10.